It's pretty amazing when you think about it, the potential of cancer immunotherapy, like harnessing your body's own defenses
Yeah,
to basically wipe out tumors.
absolutely.
Yeah.
It's really remarkable how far we've come.
It is, but the thing is, it doesn't work for everyone, right?
Right, that's the big challenge right now.
Yeah, like what's the secret sauce that's missing for all those other people?
That's the million-dollar question, really.
Yeah, so today we're diving deep into this study that was in CFEL that tries to figure out a piece of that.
Yeah, this one's really interesting.
It is, and it looks at this question of why CD4 plus T cells, which you'd think would be like major players in fighting tumors,
Right.
often just don't seem to, do what we'd expect.
Yeah, they're kind of underperforming in a lot of cases.
Yeah, and the study finds that this specific type of dendritic cell has a lot to do with it.
It's all about that interaction.
Yeah. So for everyone listening, our goal today is to really break down how these different immune cells are talking to each other or maybe not talking to each other.
Right, the communication breakdown.
Yeah, within the tumor environment and how that affects whether our bodies can actually beat cancer.
There's like a whole ecosystem in there.
It is, and we want to give you like a clear picture of that.
Absolutely, and what's really cool is that this study goes beyond just theory.
Right.
They use this really advanced technique called single-cell RNA sequencing.
Ooh, fancy.
Yeah, it lets them look at the activity of individual immune cells in incredible detail.
So not just like an average of a bunch of cells.
Exactly. They can see what each cell is doing specifically.
Wow.
And they did this in both mouse models and samples from human tumors.
So like real world validation.
Exactly. Which makes the findings even more compelling.
So let's start with the basics, I guess. We always hear about CD8 plus T cells being like the main cancer killers, right?
Right. They're the ones that get all the attention.
Yeah, they're the hitmen.
Yeah. The assassins, they directly recognize and kill those tumor cells.
But this study's more about CD4 plus T cells, right?
Yeah, it's shining a light on a different kind of player.
Okay, so these are still T cells, but they're like a different branch of the family.
Exactly. And for a long time, we thought they were mostly helper cells.
Okay, supporting the CD8s.
Right. But it turns out they're a lot more versatile than we thought.
More like a Swiss Army knife.
Exactly. They're really important for what we call licensing dendritic cells.
Licensing? What does that even mean?
Well, it's basically like giving the dendritic cells permission and instructions to activate other immune cells.
So like the dendritic cells are the generals, and they need orders from the CD4s.
Kind of, they're more like the intelligence officers gathering intel, and then the CD4s telling them how to use it.
Ah, okay, that makes sense. And you said they're versatile. What else do they do?
Well, some research suggests they might even be able to directly kill tumor cells themselves in some cases.
Whoa, really?
Yeah. And there's also evidence that patients with more active CD4 plus T cells respond better to certain cancer therapies.
So they're kind of like the unsung heroes.
They're definitely playing a more critical role than we initially appreciated.
So if they're so important, why aren't they always saving the day? Why doesn't immunotherapy work for everyone?
Right, that's the big mystery, and that's what this study really digs into.
Okay.
The problem is that in a lot of tumors, these CD4 plus T cells become dysfunctional.
Dysfunctional? What do you mean?
Basically, they get exhausted, they lose their ability to coordinate a strong immune response.
Like they're burned out.
Exactly. And even some of the checkpoint inhibitor drugs that we use now, which work by taking the brakes off the immune system.
Right.
They don't always seem to be able to rescue those exhausted CD4 plus T cells.
So even if you release the brake, the engine's still busted.
Yeah, it's a tough problem.
So where do these dendritic cells fit in? You mentioned them earlier.
Right, dendritic cells, or DCs, they're like the key messengers between the tumor and the rest of the immune system. They're the ones that travel to the tumor, pick up tumor antigens, and then carry those back to the lymph nodes.
Like little spies?
Exactly. And in the lymph nodes, they present those antigens to the T cells, essentially showing the T cells what the enemy looks like.
So they're like training the T cells to recognize the tumor.
That's a good way to put it.
And you said there are different types of dendritic cells.
Yeah, the main ones we talk about are CDC1 and CDC2.
Okay, what's the difference?
Well, for a long time, we thought it was pretty simple. CDC1s activate the CD8 plus T cells, the killer T cells.
Right.
And CDC2s activate the CD4 plus T cells, the helper T cells.
Okay. Division of labor.
Right, but this study really challenges that simplistic view.
Okay, in what way?
Well, they focus specifically on the CDC2s, and they ask this question, what specific roles do different subsets of CDC2s play?
Subsets. You mean like there are different types of CDC2s?
Exactly. It's not just one homogeneous group.
So they're like specialized units within the CDC2 team.
Right. And this is where the single-cell RNA sequencing comes in.
Right. The super detailed analysis.
Exactly. They looked at the myeloid cells from tumor-draining lymph nodes in mice.
Myeloid cells, that's a broader category, right?
Yeah, dendritic cells are a type of myeloid cell, but there are others too.
Okay.
And what they found is that there are actually at least 10 distinct myeloid cell populations in those lymph nodes.
10. That's a lot more than I expected.
Yeah, it's a really complex ecosystem. And when they zoomed in on the migratory CDC2s,
The ones that travel from the tumor to the lymph nodes.
right, they found something really interesting. There were 2 distinct subgroups within the CD11B plus CDC2 cells.
Okay, hold on. CD11B plus, what does that mean?
It's just a marker on the surface of these cells, a way to identify them.
Okay.
So they found these 2 subgroups based on whether they express another marker called CD301B.
So you have like MCD301B negative cells and MCD301B positive cells.
Exactly, and they confirmed this difference using other markers too, like CD9.
So they really nailed down that these are 2 distinct flavors of CDC2.
Exactly, and importantly, they found that both of these subtypes were carrying tumor antigens.
So they were both interacting with the tumor. Exactly. Which means they're both potentially involved in activating those CD4 plus T cells.
Okay, so we've got these 2 subtypes of CDC2. They're both picking up intel from the tumor and heading to the lymph nodes.
Right.
You'd think that would lead to a strong immune response, but you mentioned a priming problem earlier.
Right. That's where things get a little tricky.
So what's the problem?
Well, they tested how well these different CDC2 subtypes could actually activate naive CD4 plus T cells.
Naive meaning they haven't encountered the tumor yet.
Right, so they're like fresh recruits.
Right.
And they found that both the MCD301B negative and positive CDC2s could make the T cells proliferate.
Meaning that they started dividing and multiplying?
Exactly, but they weren't driving those T cells towards a fully activated effector state.
Effector state, that's like the T cells being ready for battle, right?
Exactly. They're looking for specific markers on those T cells that show they're really fired up and ready to kill.
Okay.
Things like ICOS, which means they're getting activated, but low levels of PD-1, which is an inhibitory marker, so they don't want them to be told to stop.
Right. And this combination of markers usually points to a Th1-like phenotype, which is really good at fighting tumors.
Okay.
But the CDC2s weren't doing a good job of creating that phenotype.
So it's like the T cells are getting recruited, but they're not getting the right training or equipment?
That's a great analogy.
Yeah.
And to highlight how bad this was, they compared it to what happens during a viral infection. In a viral infection, you see really strong activation of CD4 plus T cells into that effector state.
So it's like the immune system knows how to do it right, but something's going wrong in the tumor environment.
Exactly.
So we have these CDC2s. They're supposed to be turning on the CD4 plus T cells, but they're not doing it right.
Right.
What else could be going on in there?
This is where regulatory T cells come into play, or TREGs for short.
TREGs, those are the ones that suppress the immune response.
Yes, they're like the peacekeepers, making sure the immune system doesn't go overboard and attack healthy tissue.
Mm-hmm.
But in the context of cancer, they can be a real problem because they can suppress the immune cells that are trying to kill the tumor.
So they're like protecting the enemy?
Kind of. And the researchers found a correlation between the presence of those CD11B plus CDC2 cells and TREGs in the tumor.
So maybe the TREGs are interfering with the CDC2s.
That's what they wanted to find out. So they did experiments where they depleted the TREGs.
I got rid of them.
Exactly. And what they saw was really amazing. The tumors started to get rejected. And this rejection was dependent on CD4 plus T cells that had been primed in the lymph nodes.
Meaning the T cells that were trained by the CDC2s?
Right. They used a drug called FTY720, which traps T cells in the lymph nodes to prove this.
So if the T cells couldn't leave the lymph nodes, the tumors didn't get rejected.
Exactly. Which means those lymph node primed T cells were essential.
Wow. So getting rid of the TREGs made a huge difference.
It did. And it wasn't just about the T cells. They also looked at what happened to the CDC2s themselves.
Okay, what did they find?
Well, when they got rid of the TREGs, they saw an increase in the migration of both those MCD301B negative and positive CDC2s to the lymph nodes.
So more of them were making the trip to train the T cells.
Exactly. And they were carrying more tumor antigen, too.
So they were better equipped to show the T cells what to attack.
Right. And on top of that, the CDC2s from those TREG-depleted mice were much better at inducing those T-cell activation markers.
The ICOS and PD-1 ones we talked about earlier.
Exactly. They were expressing more of the co-stimulatory molecules like CD80 and CD86.
Those are like the go signals, right?
Yeah, and they were also producing more chemokines, which attract T-cells.
So they were basically sending out a much stronger call to arms.
Exactly. And the end result was that you got more of those ICOS positive, PD-1, low, Th1-like CD4 plus T-cells.
The ones that are really good at fighting tumors.
Exactly, both in the lymph nodes and in the tumor itself.
That's amazing. So it seems like the TREGs are really putting a damper on these CDC2s.
Yeah, they're preventing them from doing their job properly.
Which makes you wonder if we could target those TREGs therapeutically.
That's exactly what they looked at next.
Okay, what did they find?
Well, they looked at this antibody called anti-CTLA4.
Which is already used to treat some cancers, right?
Exactly, and it's known to deplete TREGs in the tumor.
Okay.
And what they found is that this antibody also increased the migration and those functional markers on CDC2s.
Just like when they directly depleted the TREGs.
Exactly, which suggests that some of the effectiveness of anti-CTLA4 might be due to its effects on CDC2s.
That's a really cool connection.
Yeah, and they even looked at combining anti-CTLA4 with a cancer vaccine called GVAX.
Okay.
And they found that this combo led to even better CDC2 activity and anti-tumor responses.
So boosting the CDC2s along with depleting the TREGs?
Exactly. And these effects were dependent on the presence of CDC2s.
Meaning if you got rid of the CDC2s, the COMER didn't work.
Right, which really highlights the importance of these cells.
This is all really promising, but it's all in mice, right?
Right, so the next question is, does this hold true in humans?
Yeah, what did they find?
Well, they did single-cell RNA sequencing on human tumor-draining lymph nodes.
Yeah.
They found a similar level of diversity within the BDCA1 positive CDC2 population.
BDCA1 positive. That's the human equivalent of CD11B+, right?
Exactly. And they found subsets that were analogous to the mouse MCD301B negative and positive populations.
So the same basic subtypes exist in humans.
Yeah, which is a good sign that the underlying biology might be similar.
And did they look at any actual human cancers?
Yeah, they analyzed head and neck tumors, and they found that patients with a high abundance of those BDCA1 positive CDC2 cells and low levels of TREGs in their tumors.
Oh, okay.
They had more of those beneficial ICOS positive PD-1, low CD4 plus T cells in their tumors.
Yeah.
And they had better progression-free survival.
So they lived longer without their cancer getting worse.
Exactly, which really strengthens the link between these CDC2s and clinical outcomes.
That's really exciting.
Yeah, and they even reanalyzed data from melanoma patients. And they found some evidence that the abundance of those BDCA1 positive CDC2 cells alone might be able to predict how many CD4 plus T cells are present.
And maybe even how well patients respond to anti-PD-1 therapy.
Right, which is another type of checkpoint inhibitor.
So potentially these CDC2s could be a biomarker to help us choose the right treatment.
Exactly.
So to sum it all up, it seems like these CD11B plus CDC2 cells in mice and their BDCA1 positive counterparts in humans are really crucial for getting those anti-tumor CD4 plus T cells fired up.
Yeah, they're like the key to unlocking that immune response.
But their activity is being suppressed by these TREGs in the tumor environment.
It's like they're being held back.
And if we can figure out how to overcome that suppression, we might be able to make immunotherapy work for a lot more people.
Absolutely. That's the big takeaway here.
So we've covered a lot of ground today for everyone listening. What are the key things you want them to remember?
The main points are that we've identified these specific dendritic cells, the CDFNB plus CDC2s and their human equivalents, as the main drivers of those anti-tumor CD4 plus T cell responses.
Okay.
We've learned that they're not working as well as they could because they're being suppressed by TREGs.
Right.
And maybe most importantly, we've seen that if you get rid of those TREGs, you can unleash the power of those CDC2s and get a much stronger immune response.
It's like taking the handcuffs off those dendritic cells.
Exactly.
And I think the really mind-blowing thing here is that it's not just about getting T cells into the tumor.
Right.
It's about making sure they're properly activated right from the start in the lymph nodes.
That initial priming is crucial.
Yeah, and if we can improve that priming step by targeting those CDC2s or the TREGs or both,
Right.
we might be able to revolutionize cancer treatment.
I think that's a really exciting prospect.
It is. So for all of you amazing learners out there, as you go forth and learn more about this stuff,
Yeah, keep learning.
think about this. How could we use this knowledge to develop even more personalized and effective cancer treatments?
It's a big question.
It is, but this study gives us a lot to work with, and it really highlights how important it is to understand the complex interactions between all these different cells in the tumor.
It's not just about targeting one thing. It's about understanding the whole system.
Exactly.
Yeah.
And with that, I think we'll wrap up this deep dive. Thanks for joining us.
It was a pleasure.
And keep those brains buzzing.